Hey everyone, in this video we're going to go over the process of gluconeogenisis. So what I've drawn here is actually a figure that you've seen before. This is actually the pathway of glycolysis. I've actually just started here with glucose 6-phosphate, and recall that glycolysis converts G6P down through a series of steps, ultimately to pyruvate. Now in the process of gluconeogenesis, this is going to be sort of an uphill battle because it's going to require energy. Recall that catabolic processes are downhill in energy. They produce energy compounds like ATP. But in order to build a molecule, so an anabolic process, we're going to take 2 pyruvates, and ultimately we'll be making 2 glucose molecules. All right, now, this step right here, this is in one of the previous videos I talked about. This was a regulation point right here. So the energy that's released from phosphoenolpyruvate going to pyruvate, it's almost impossible to go directly backwards. So in order to sort of get around this energetic hurdle, and notice I've written this as a one-way arrow, the 2 pyruvate molecules are actually going to be converted to oxaloacetate. And there will be 2 of those. Now recall that pyruvate is a C3 compound, so there's 3 carbons in that. Oxaloacetate is a C4 compound. And what's going to happen is the enzyme involved in this, called pyruvate carboxylase, so that's our enzyme, is going to take 2 ATP plus 2 CO2s and convert that to 2 ADPs. So what's going to happen is this enzyme is going to be sticking on the carbons of carbon dioxide onto pyruvate to make oxaloacetate. All right, and that does require 2 energy equivalents of ATP to do that. So it sort of makes this little bit more energetic molecule called oxaloacetate. Next, oxaloacetate is going to be ultimately converted to phosphoenolpyruvate through the action of the second enzyme in this control point right here. And that enzyme is called Pep Carboxykinase. Okay, so that's the enzyme. It's often abbreviated as PEPCK. And this one is going to require 2 GTP molecules, and it's going to convert that to 2 GDPs plus 2 CO2s. So the CO2 that was put on in the first step is ultimately going to be coming off in this second step. And notice it does require 2 equivalents of ATP energy. So in biochemistry, we essentially equate GTP energetically to an ATP as well. So just to get around this one step, we use a total of 4 ATP equivalents just to go from pyruvate to phosphoenolpyruvate. Okay, so that is the first sort of control point that is definitely different in gluconeogenesis. Okay, now going in the upwards direction, there are a couple other things that we really need to put on here. First, in this step right here, 2 ATPs are converted into 2 ADPs right there. So we use another 2 equivalents of ATP. This is just a reversal reaction of the glycolysis enzyme. Next, in this conversion here, there is a loss of NADH. So we're going to take 2 NADHs, and these will be converted back to NAD+. Okay, so it also, in order to build molecules of glucose from pyruvate, it's going to require another energy molecule in the form of NADH. Lastly, there are these 2 steps up here that are going to involve phosphatase enzymes, and these are going to be different than the glycolysis enzymes. So right here, the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate, this will involve the use of water to simply liberate an inorganic phosphate, which we just call PI. And the name of that enzyme is fructose 1,6-bisphosphatase. Okay, now notice there's no ATP or any energy molecule involved. This is simply a hydrolysis of the inorganic phosphate. And then lastly, if the cell does receive signals that ultimately make the cell want to liberate its glucose so that it can be pushed out into the blood, this last enzyme is going to be involved, which will convert glucose 6-phosphate into glucose. Okay, and that enzyme also is a phosphatase. All right, so we'll use water. Just going to cleave that to give it inorganic phosphate. And the name of that enzyme is just simply glucose 6-phosphatase. Now, you may recall I've mentioned several times that cells are quite greedy when it comes to glucose. So not all cells even have this enzyme present. Certainly brain cells do not even express that enzyme because brain cells absolutely require glucose as a fuel. So some tissues don't even produce this enzyme. All right, so again, this step right here, this step right here, and this step right here, those are the 3 regulatory points that basically allow the process of gluconeogenesis to be turned on while glycolysis is turned off, and vice versa. You wouldn't want to have the 2 pathways going at the same time. So just to demonstrate that, let's just add in some metabolites here going in the downward direction. So if I go through glycolysis, just to refresh our memories, in this first step, I use an ATP. Actually, I use 1 ATP here. 1 ATP. In the second step, I use 1 ATP. Okay, down here, I end up getting out 2 NADH. And here I get 2 ATP. And let's see, and then ultimately here I get 2 ATP. So on this left-hand side of the figure, in the downward direction of glycolysis, my net gain would be plus 2 ATP for glycolysis. And I would have plus 2 NADH. Okay, so that's review. Over here, in the upwards direction, ultimately there's going to be a net loss of energy. Let's just summarize that over here. So gluconeogenesis, net loss, we have minus 6 ATP equivalents. Let's count them up here. We got minus 2 right here. We have minus 2 ATP equivalents here. And we have minus 2 ATP equivalents here. So a total of minus 6. Remember that GTPs are just counted as ATPs as well. And then we also have minus 2 NADHs. So having this figure now in hand, I would highly recommend that you actually use this combined figure of both glycolysis and gluconeogenesis to study both pathways. And the last point I want to make about this, what would happen if a cell had both of these pathways going on at the same time? Well, let's think about that. So if we had glycolysis going on, we know that our net gain would be plus 2 ATP. But if we had gluconeogenesis activated at the same time, that would lead to a net loss of minus 6. So if both pathways were cycling back and forth, ultimately, for every time we went through one cycle, we would ultimately lose minus 4 ATP. So this illustrates a point or a type of opposing pathways called a futile cycle. Cells do not like futile cycles. They do not like opposing pathways to be turned on at the same time because ultimately it makes the cell lose energy. So at these key control points, here, here, and here, these enzymes that are different between the two pathways are actually going to be regulated so that one pathway can be on and the other off. Okay, so this figure, again, one that I highly recommend you sort of memorize, put them both together, glycolysis and gluconeogenesis, and then be aware of this issue of futile cycles. All right, I hope that helps, and good luck.